Harvest is a really important day because that's when we are taking the product of our 10 plus days of work and generating a dose for the patient. Key things on Harvest are the number of cells that we have in the culture and that we have at the end of the process after we've completed our washing and preparation for freezing. Just as the isolation, we have a cryoprotectant solution that we use with the cells to protect them through the freezing process. And we're also looking at our CAR expression again on day 10 to verify what percentage of our cell population is expressing the CAR, which will help us determine dose.
So the MACI is autologous cultured chondrocytes. We actually grow the chondrocytes up and then we're going to seed them onto this porcine collagen membrane. Chondrocytes are what's termed an adherent cell population, so they're going to bind any surface in their cell culture environment. When they bind to this membrane, we then ship the membrane with the bound chondrocytes to the physician. The physician can then cut the membrane to size and implant it into the area of the defect for a perfect match. The collagen is a type 1, type 3 of a porcine or a pig derivative, and it's resorbable. So as it basically becomes the medium to deliver the cells into the area of the defect for chondral, osteochondral applications, and the membrane will break down. And while it breaks down, the patient's own chondrocytes from the membrane have the opportunity to ingress, grow into the defect, and basically rebuild cartilage in that space for the patient.
So, you should now have a broad understanding of the steps needed to manufacture a cell therapy. But of course, it's not enough to be able to manufacture that product once or even a handful of times for a few patients. We also need to ensure that we can manufacture an equivalent product no matter how many batches we manufacture. And this is particularly challenging for autologous cell therapies because there's inherent biological variability and each patient is a separate batch. So to minimize that product variability, we have significant effort spent on ensuring that quality is built in to the manufacturing process operations. For complex biologic products like cell therapies, it's often said that the process is the product. And that's because with such complexity, it's not currently possible to understand how seemingly small changes in the manufacturing process might impact the quality of a cell therapy or to fully characterize that final product and predict the clinical impact of what's really an unanticipated change in a product attribute. So to ensure consistent product quality, manufacturers tightly control the process, and the goal there is to minimize the variability of cell therapy characteristics that cannot be measured directly. But of course, further, no amount of testing can salvage a poorly made product.
So quality, you can break it down into several different subdisciplines. It's quality control testing of both the final product and the raw materials. Validation activities to demonstrate that what you think a process will do is actually what it does. Also, quality assurance and quality systems. So most companies, particularly cell therapy companies that are regulated by the FDA, will have a quality system, which are the SOPs and the structures that you follow in order to produce your product and monitor it.
Now, as you just heard from Mike Halpin, cell therapy manufacturers employ what we call a quality system to both control and assure the manufacturing operations and the final product quality. Fundamentally, a cell therapy manufacturer needs to document, first, what they want to do before they do it by defining the process. Second, do exactly what they said they were going to do. And third, be able to prove it. So let's look at each of these aspects in turn and see how they contribute to the overall quality control of cell therapy manufacturing operations. So first, you need to say what you're going to do before you do it. And to do that, you need to define the manufacturing process By this, we just mean the steps that the operators will take to perform that process, and then the limits within which that process needs to operate. This includes setting specifications for the raw materials that are inputs for that process, including the cells. Now, proving that a defined manufacturing process performs as intended is formally known as process validation. Let's hear about that more from Humberto Vega.
Process validation is a complex approach to demonstrate and to document that a manufacturing operation performed as intended, as designed. When we talk about process validation, we talk about three different stages. One is a developmental stage, where a process is designed, the proof of concept is developed, data is collected, and that process is safe to do what is intended to do. In the case of CAR-T, that you can take T cells and transform them into a modified T cell with a particular antigen receptor. The second stage, which is the PPQ, or Process Performance Qualification stage, is where the final process is tested against some particular acceptance criteria to show that the process is working as intended and will be able to be repeated batch after batch. And you end with a product that is the same every single time.
The final process and the steps that the operators need to take to perform that process also need to be clearly documented. And we do that in standard operating procedures, or SOPs, and master batch records, or MBRs. Now, these SOPs and MBRs are really just the instructions that operators must follow to ensure the manufacture of a high-quality final product. Of course, it isn't sufficient to just say what you're going to do. You need to follow through and actually do it. And so for that, training and continuous learning and operator assessment are really just key tools that we use to ensure the manufacturing process is performed the same way every time for every batch for every patient. So staff are trained on the manufacturing steps, on the proper use of personal protective equipment or PPE, on the procedures to prevent cross-contamination, and on any other considerations down to the flow of people and materials through the facility.
To go through the rigors of our gowning qualifications, our aseptic technique qualifications, where they demonstrate with media designed to grow bacteria, that's basically acellular, they demonstrate I can perform a simulated process in the biosafety cabinet and not introduce bacteria or microbial contamination. Then we go through the qualification on the various steps in the manufacturing process. So I think 6 months is a good time period just for people to really become qualified, acclimated, and begin performing at an independent level.
Next, you need to prove, both to yourself and to the regulators, that you perform the manufacturing process as it was intended. And so to do this, you have to demonstrate that you had control over the raw materials, the equipment, the facility, the environment in that facility, and the manufacturing process itself. So first, the raw materials. The raw material quality has to be documented prior to use in the manufacturing process. And so suppliers will be asked for certificates of analysis, or COAs, for those products. Some manufacturers will perform their own raw material testing on top of that, either to confirm the supplier's test results or to ensure that they meet internal performance specifications at the facility. Next, controls are put in place to ensure an appropriate manufacturing environment. For example, the water and the air entering the facility are treated to remove bioburden and to remove particulates, and the surfaces are sanitized and sterilized. Further, that environment is tested and monitored to ensure that these procedures are both followed and continue to be effective. Finally, you need to prove that you had control over the manufacturing process itself. And this is, again, accomplished through documentation and testing. Operators will document specific manufacturing operations at the exact time they're conducted and will record and confirm all of the relevant information, such as the raw material lot numbers or the actual process conditions or parameters. And recording these little details is very important because those recorded values will be compared to the expected values that are listed in the SOPs and the MBRs. We do that to confirm that the process was executed as designed for satisfactory process performance. Now, along the way, samples will be taken throughout the process, either at predetermined times or between specific operating steps. Some of those samples will be saved for later evaluation, but many of those samples will be tested during the manufacturing process so that the quality attributes that are required for manufacturing are known and maintained. And at the end of the process, lot release testing will be performed. This is to confirm that the product meets its target specifications. And those specs include product identity, purity, and the absence of contamination to show safety, as well as measures of function. And these are together known as the critical quality attributes, or CQAs, that we discussed earlier. Now, all of this testing and documentation is compiled into a record for each batch of product. These batch records are then evaluated for any deviations or differences before that final product is released. Finally, as we mentioned earlier, you need to ensure that the manufacturing process operates the same way every time. In other words, we must continuously verify that your process performs as expected.
The last stage of process validation, which is a continued process verification stage, is where, as you manufacture additional lots, you continue to monitor those parameters and those quality attributes and confirm that in each batch they come back as intended, as validated, as designed and developed.
Now, as you can see, each element of this quality system works in concert with the others to ensure that the manufacturing process operates as intended and produces the desired product. Of course, that desired product is a safe and effective cell therapy product for every single patient, every single time.